Evening, people. Good evening. Hello. How are you? Can you hear me clearly, people? Can you hear me clearly? Is the sound level okay? What do you message dan lamai? Can you hear me clearly? Yes. Santosh says yes. Arenade. Right, students. We have to discuss paper 4 today. Paper 4. Can somebody uh, paper 4? Paper 4 today. Right. Paper 4. Find paper 4, people. Hope you have the paper with you. Paper 4. Paper 4. Find paper 4. All of you have paper number 4 at the moment. I think you all have it. I have a small difficulty of sending that the paper 4 to the WhatsApp chat. Can someone send me the paper 4? That's a big help. If somebody is having paper 4 PDF, send it to me. Now I'll share it. I can't transfer it from one computer to the other. I don't know how to do it. Okay. All of you have paper 4 right now, people? There is no necessity of paper 4. Giving PDF, no need, ne? No need, ne? Then we'll continue with the discussion. Then let's continue. Please send me also in the website. It's not there. No, it's available in the website. Why do you say not available in website? It's available in website. I uploaded it to the website. Shall I send it to the group, people? I can send it to the group. I'll send it to the class group. Huh? I'll send the paper to class group. Pick it from the class group. And yeah, it's in the class group now. Rishma. Rishma. Can you find it from the class group? How did I got it? How did it? Right. So people, first question. Correct sequence of hierarchical levels of organization of living organisms. Hierarchical sequence, correct order of hierarchical sequence of organization of living matter. Correct sequence. What is the correct sequence, people? I'll look at that. Okay. Right. Correct sequence of hierarchical levels of organization of living organisms. Cell, organ system. Again, anime buttons stick deal anime. Black no. ඊළඟ මේ මේකට කොහොමද මේ ටෙලිග්‍රෑම් නෑනේ මේකේ මට දාගන්න බෑ ෆයිල් එකක් මේකට මේ ටෙලිග්‍රෑම් දාන්නකෝ අඩු ගන්න මේකෙන් මේකට ෆයිල් එක ට්‍රාන්ස්ෆර් කරන්න බෑ නෑ වැඩ නෑ ඔයාලට කියන්න බෑ මේක මේ අර මෙතන වෙබ් කැම් එක ඒක ඔෆ් කරලා තියෙනවා කොහොමද හරි හරි Question number 1, people. Correct sequence of hierarchical levels of organization of living organisms. Correct sequence of hierarchical organization. Cell, organ, organ system, individual. Correct sequence of hierarchical levels. Now, cell, organ, now a lot of stages are missing. A lot of stages are missing. හරිද? හරිනේ? හරිනේ. A lot of stages are missing, people. Cell is there, organ system, individual. Some steps are missing, right? Some steps are missing. So then it's not suitable as the answer. Organelle, cell, tissue, organ. This looks okay because in between steps are not missing. Others, you can see that first one, after the cell, there must be tissue. Tissue, and thereafter it should be organ. So first is not good. Tissue, organ system, species, individual is missing. Individual, population, community, biosphere. Individual, population, community, individual, population, community, biosphere. Individual, population, community, biosphere. Then biosphere, community, biosphere, මෙතන අඩුයි නේද? Ecosystem නේ. ඊළඟට biosphere, ecosystem, community, population. Biosphere, wrong නේද? So we have the answer, people. Best answer, you can go for the second one. Best answer is second. Right? Best answer, second. Hari. Answer second. Hari. Question number two. Question number two. Which of the following statement is correct regarding properties of water? Correct regarding properties of water. High surface tension is given to water molecule due to addition. High surface tension is given to water molecule by addition. Addition or by cohesion. The cohesion. Addition is wrong. Water will function as a thermal buffer in living systems due to high heat of vaporization. High heat capacity. Wrong. Solubility of solutes depends on the ionic nature. Solubility of solutes depend on the ionic nature. Solubility depends, solutes depend on the ionic nature. Solutes depends on the ionic nature. Ionic nature of water but then it's wrong. But this is not a very clear statement. High heat of vaporization of water helps to maintain body temperature at constant level. Yeah, that's not wrong. When temperature of water falls, the density of water always increases. When temperature decreases below 4 degrees Celsius, density decreases. It doesn't happen always. Always word is wrong. Right? So you got the answer now. Answer we can take, people, as the fourth one. Answer is fourth. Question number 3. In which of the following compound monomer is a ketose? What is your answer? Monomer is a ketose. Glycogen, what is the monomer? Glucose. It's not a ketose. Glucose is an aldose. Hemicellulose, pentose sugars. No. Chitin, glucosamine. No. Inulin, correct. Fructose. Fructose is a ketose. You learn that. And pectin. What is the monomer of pectin? Can someone tell me the monomer of pectin? What is it? What is monomer of pectin? Galactronic acid. Very, very good. Galactronic acid. Very good. Danisha. Right. Sama. Galactronic acid. Good. Right. Next one, people. Question number 4. Question number 4. What is the molecular weight of cellulose molecule made up of 500 glucose units? How do you work it out? How do you work that out? How do you find it out? Glucose molecule, but what is the molecular weight of glucose? Can somebody tell me what's the molecular weight of glucose? What is the molecular weight of glucose? 180, ne? 180 into 500, ne? 180 into 500. What's the value? 180 into 500. What's the value? Can somebody calculate and tell me? 180 into 500. Come on, I can calculate, but just take time. What's the value? Right? Come on. And you have to deduct how many water molecules are eliminated here? How many water molecules are eliminated? You're not going to tell me. I'll find it then. 180 into 500. 90,000. 90,000. Minus how many water molecules you're going to reduce? Yeah. We answer, say C6H. H10. That's correct, ne? H10O5 multiplied by 500. I don't think that's suitable. Now, we answer say that C6H10O5 multiplied by 500. No. Why? You know the reason? This is it. This is the reason. Now, let's take 3. How many water molecules come for 3? How many water molecules come for 3? 2, ne? How many water molecules come for 3? 2. Then how many water molecules come out for 500? 499. 499 into 18, ne? Into 18. H2O 18. So 499 into 18. Roughly, I think the value comes closer, ne? So what are you going to do? 18 into 499 minus 90,000. 81,000. 18. We have the close answer. We have exact answer. Ne? Right? So, Vihansa, did you get the same answer? 81,018. 81,018. Did you get that exact number? No, ne? That's because this has to be 499. Ne? So that's the reason. Because every 2, 1 water molecule, every 3, 2 water molecule, 499, you deduct, then for 500, 499. Right. Next one, people. Which of the following is not a function of monosaccharides? Which of the following is not a function of monosaccharides? Not a function of monosaccharides. Not a function of monosaccharide. Acting as energy source. Acting as energy source. Not a function of monosaccharide. We got this one as the answer. Which of the following is not a function of monosaccharide? Acting as energy source, correct? Function, ne? Acting as component of nucleotides, correct? Acting as carbon dioxide acceptors, monosaccharides? Carbon dioxide acceptor? Danisha. Nisha. Is there any monosaccharide acting as a carbon dioxide acceptor? Any monosaccharide that you know act as a carbon dioxide acceptor, people? Do you know any monosaccharide reacting as a carbon dioxide acceptor? Come on. And then folks, good, Santosh. RUBP, Shah. RUBP, that's correct. So acting as carbon dioxide acceptor also correct, ne? Acting as carbon dioxide acceptor, correct. Then contribute for synthesis of macromolecules, correct. Acting as storage component of cell. Which of the following is not a function of monosaccharide? Directly act as storage component, wrong, neither. Sugars like glucose are not storage materials. They can convert into relatively less soluble material, right? Monosaccharides are not storage, neither. They are readily utilizable. They don't store it. They store it either starch or maybe sometimes people sucrose. Ne? Sometimes sucrose, but not directly as the starch. Ne? Not directly as glucose. Ne? So we'll go for the fifth one as the best answer. Fifth one as the best answer. Question number 6. Question number 6. Question number 6. Which one of the following correct regarding triglycerides? Correct regarding triglycerides. Triglycerides correct kahanne. Right? Triglycerides. Large biological molecule, macromolecule, polymer. Triglyceride, large biological molecule. False. Neither. Triglyceride is not a large biological molecule. It is not a macromolecule. It is not a polymer. Triglyceride. So, false, false, false. Do you have that? False macromolecule. Which of the following regarding triglyceride? False, false, false is my answer. False, true, false. False. Wait. I'm wrong. Now, this is the reason. Ne? Now, it doesn't say large biological molecule. That and the macromolecule are two different things. Ne? That is how we took in the resource book. Ne? It is given like that. Ne? So, large biological molecule, large, but it's not a macromolecule. Ne? So, true, false, false. True, false, false should be the answer. Ne? True, false, false. True, false, false should be the answer. Right? Right? Selvanathan says large biological molecule, but not considered as a polymer or macromolecule. Very good. Right? I forgot that point. Right. Question number 7, people. Backbone of amino acid consists of. Backbone of amino acid consists of. Backbone of amino acid consists of. Only R group. Only R group. Now, what actually present in the backbone? Not the R group, neither. Amine group, carboxyl part, the central carbon atom, and the hydrogen group, neither. They are only R group. So, backbone wrong. The carboxyl group of an amino acid. No. Carboxyl group, amino group, and hydrogen atom. Correct. With the central carbon atom, only carbonyl group. These also wrong. They are also wrong. So we can go for people. Third one as the answer. Third one is the answer. Question number 8. Which of the following statement regarding light microscope is correct? They are asking correct statement regarding light microscope. Cannot observe by naked eye. Which of the following statement regarding light microscope correct? Cannot observe, wrong. Actual color of the object cannot be seen, wrong. You can see, less resolution power than the electron microscope. Yeah. And then heavy metals are used, no. Strong magnets are used, no. So we got the answer. Answer is third. Answer is third. Very good. Right, moving to next page. Next page. Next page. Right, next page. Right, select the correct statement about eukaryotic nuclear division. Correct statement regarding eukaryotic nuclear division. Right, correct regarding eukaryotic nuclear division. Nuclear envelope is fragmented and centrosomes are located in the metaphase plate during the pro-metaphase. Nuclear envelope is fragmented and the centrosomes are. Correct statement regarding nuclear division. Nuclear envelope is fragmented and centrosomes are located. Nuclear envelope is fragmented and the centrosomes are located in the metaphase plate during pro-metaphase. What do you say about that, people? Select the correct statement. Correct statement. Nuclear envelope is fragmented. Prometaphase, we know people, nuclear envelope is fragmented. And centrosomes are located in the metaphase plate. That is wrong, neither. Centrosomes are located in the metaphase plate. Centrosomes, centromeres, centromeres are located in the metaphase plate during the prometaphase. That is wrong, ne? Nuclear fragment in the prometaphase, this part is okay. This is correct. But this part is wrong, ne? That part is wrong. Kinetochore microtubules move chromosomes back and forth during the anaphase. That is wrong, neither. Prometaphase. And then both chromatids of one chromosomes are attached to. Now, this question is regarding people eukaryotic nuclear division. Just ask about the eukaryotic nuclear division. Not mitosis, not meiosis. Ne? So we have to keep that in mind. Na? Kinetochore microtubules move chromosomes. Okay. Both chromatids of one chromosome are attached to kinetochore microtubules from one pole. One pole in prophase. Both chromatids of one chromosomes are attached to kinetochore microtubules from one pole in prophase. Wrong, neither. Prophase. Both chromatids of one chromosome are attached to the microtubules from one pole in prophase. People, it talks about the nuclear division, neither. Not mitosis. Right? Not mitosis, not meiosis. Select the correct statement. Right? Both chromatids of one chromosomes are attached to kinetochore microtubules from one pole in prophase, metaphase. Now, how about people in meiosis? If you consider meiosis, both chromatids of one chromosome are attached to kinetochore microtubules from one pole in prophase. Metaphase that happens in meiosis, ne? Metaphase that happens in meiosis. We'll leave this third statement, neither. Fourth one, check karan sister chromatids of one chromosome move to same pole in mitosis. Sister chromatids of one chromosome move to the same pole. Wrong, neither. Generally, non-identical four daughter nuclei are formed at the end of the anaphase 2. Genetically non-identical. That is correct, people, for meiosis. Ne? Question says just the nuclear division. Genetically non-identical four daughter nuclei are formed at the end of the anaphase 2. Anaphase 2. Telophase 2. Ne? Telophase 2. Anaphase 2 segregate. Ne? Segregate. Ne? And then, right? Telophase 2 is more correct. Ne? Telophase 2 is more correct. Right? What is your answer? At the end of the anaphase, end of the anaphase. Now, end of the anaphase is what, people? End of the anaphase is the beginning of end of anaphase. They have migrated to the opposite poles, ne? Migrated to opposite poles. And telophase, the four nuclei, and telophase followed by the cytoplasmic division, ne? I think end of telophase 2 is better, ne? So what's the answer then? What is the answer? Little different question, ne? Students say fourth kilat kina samarakkai. Sister chromatids of one chromosome move to same pole in mitosis when the bani. Right? Mitosis when the make a wrong. I think fifth one also wrong kila mama hitandi. Both chromatids of one chromosomes are attached to kinetochore microtubules from one pole in prophase. That does that happen in people? Does that happen in? Does that happen in? Does that happen in? Let's take people. Does that happen in? Can you take that happen in meiosis? Let's check the resource book quickly, neither. Right? Where is my resource book? Here it is. Right, we'll check that in the resource book. Fine. Look at this. Right. Let's take this one. Meiosis anaphase. Meiosis anaphase. Now look at this, people. This is meiosis 2 prophase, meiosis 2 anaphase, meiosis 2 anaphase, and then this is telophase. Only the nuclear envelope is going to reform. Ne? So that statement is wrong. How about this? How about this? Meiosis prophase. Meiosis 1 prophase. Let's read the text, people. Meiosis 1 prophase. Prophase 1. Prophase 1. DNA molecules are non-sister chromatin paired homologous chromosome break, exchange, and rejoin. This process is called crossing over. Chiasmata. And then nuclear breaks, chromosomes move towards the opposite pole, forming spindle. The kinetochore of each homologous attached to microtubules. This part. That happens before metaphase. Ne? That happens before metaphase, right, people? Huh? So, look at this. Look at this. Okay. So, we can take this statement. Third one is correct. Third one is correct. So, the answer is third. It's little tough question. Tough question. Answer is the third one. Right. Question number 10. Question number 10. Right. The fluid mosaic model described in the structure of the plasma membrane. The fluid mosaic model described in the structure of the plasma membrane, which of the following component is most responsible for the selective permeability of the plasma membrane? Selective permeability of the plasma membrane. Cholesterol molecules, phospholipid bilayer. Which of the following component is most responsible for the selective? Phospholipid bilayer, peripheral proteins, glycoprotein, cytoskeletal filaments. Cytoskeletal filaments. What do you think as the answer here? What do you think? Cholesterol molecule can't be. Ne? Cytoskeletal filament also can't be. Glycoprotein is important for the recognition. Ne? Glycoprotein recognition. Peripheral proteins are working like enzymes and all. So the best answer is this. Best answer is that. Phospholipid bilayer. Question number 11. Which of the following structure within the nucleus is responsible for the production of ribosomal subunits? Ribosomal subunits. Which one is important for formation of ribosomal subunit, people? Nucleolus, right? So we'll take second one as the answer. Right? 11th second loop. Right? That's good. Students are with me and they are answering questions before me. Ne? That's good. Question number 12, people. Question number 12. Cilia and flagella are essential for the cell movement. Cilia and flagella essential for the cell movement. Which of the following is main structural feature that distinguishes cilia from flagella? Cilia, flagella, structural difference. Cilia are longer and fewer in number. Wrong, neither. Can't be fewer in number. Ne? It's like numerous. Cilia move in wipe-like motion while flagella move back and forth motion. Weep-like movement. No, neither. We didn't say anything like that. Cilia involving in sensing environmental signals while flagella involving in cell division. No, we didn't say anything like that. Cilia are shorter and more numerous, while flagella are longer and usually fewer in number. Cilia are shorter and more numerous. Flagella are longer and usually fewer in number. That's good. More numerous. Right? And cilia are found only in prokaryotic cells, while flagella are found in eukaryote. Wrong, neither. So we got the answer. 12th one, people, answer is 4th one. Question number 13. Question number 13. Question 13. Following are few events that take place in meiosis. Once again, meiosis question. What's your answer? Can you tell me the answer first? Your answer first, Santosh. Right? Following are few events take place in meiosis. Two haploid cells are formed. Separation of sister chromatids. Separation of sister chromatids. Alignment of chromosomes in metaphase plate. Synapsis. Chromosomes with sister chromatids move to opposite poles. Which of the following shows the correct sequence of the above events in meiosis? Correct sequence of above events in meiosis. Right. Following a few events take place in meiosis. Two haploid cells are formed. That you can't take as initial, neither. Separation of sister chromatids. Alignment of chromosomes at the metaphase plate. Synapsis. Chromosomes with sister chromatids move to opposite poles. Synapsis, first one. D first, ne? D first. Chromosomes with sister chromatids move to opposite poles. Sister chromatids, that happened last, neither. Alignment of chromosomes. Ooh. Alignment of chromosomes at metaphase plate. Alignment of chromosomes at metaphase plate. How about that as the second one? Inang DC. Two haploid cells are separation of sister chromatids. Two haploid cells are formed. Chromosomes with sister chromatids move. Chromosomes with sister chromatids move to opposite poles. How do you take that? DC correct, ne? DC correct. Chromosomes with sister chromatids move to opposite poles. That happens in anaphase 2, ne? With sister chromatids, chromosomes with sister chromatids are moved to opposite poles. That is number 3. So DCE. 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 It was a two haploid cells are formed. Two haploid cells are formed at the end of the meiosis 1. And also it happens at the end of the meiosis 2. Separation of sister chromatids has to happen, neither. So DCE, DCE. Now what is the answer with DC? DCE, 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 DCE. Where is DCE? DCE? And what's your answer? What is your answer, people? DCE. We don't have that, neither. It got to be DCE, ne? DCA. You can't take DCA. DCA can't be, ne? Chromosome with sister chromatids move to opposite poles. Right? Chromosomes with sister chromatids move to opposite poles. Can it be? Right? Fine. DCE. How about DCA? DCA. Two haploid cells are formed. DCA, DCABE. B, separation of sister chromatids. B, DCA. You have an answer for that? DCA lu. So is it DCA kilahana? DCA. Two haploid cells are D. Synapses occur first one. DC. Alignment of chromosomes at metaphase plate. Alignment of chromosomes at metaphase plate. That can happen in meiosis 1 and meiosis 2. Alignment of chromosomes in metaphase plane. It can happen in meiosis 2. Two haploid cells are formed. DC. Alignment of chromosomes at the metaphase plate. Meiosis 1 and meiosis 2. Then if you take it as meiosis 2, then you can delay it further. Nimidu says DCAEB කියලා කියනවා. So should be DCEBA. DCEBA. Is it there? DCEBA? We don't have DCE. DCE is not there. DCE. Now we'll take this. Now people, first one is D. So this one can't be. This one can't be. Right? So either has to be second, third, or fourth. DC, alignment of chromosomes at metaphase plate. Chromosomes with sister chromatids move to opposite poles. Chromosomes with sister chromatids move to opposite poles. Chromosomes with sister chromatids are moving to opposite poles. Now we have DCAEB nantino neda. DCAEB. A combination in the DCAEB. Now A can fall into people neither. Two have DCA. 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 Two haploid cells are formed. That is meiosis 1 then. This is in meiosis 1. Meiosis 1 kila pitamuko. There are two haploid cells are formed at the end. You don't see this, people? Frozen neither. Frozen, oh. Right? I think something wrong. Right? Something wrong. Now okay. We have DCA. DCA. E. Chromosomes with sister chromatids move to opposite pole. B. Separation of sister chromatids. Chromosomes with sister chromatids move to opposite poles. Separation of sister chromatids. DCA. BE venduna. DCA BE. DCA BE. Make a thino. Make a hurry. Right? Second is a possibility. Second is a possibility. What do you say? Second is a possibility neither. If you consider DCA, A, two haploid cells are formed as end of the meiosis 1, and B as separation of sister chromatids. That happen in meiosis 2. B, E. Chromosomes with sister chromatids move to opposite poles. Chromosomes with sister chromatids move to opposite pole. Chromosomes with sister chromatids. It is possible neither. Then it's wrong neither. chromosomes with sister chromatids means that definitely happen in people. Meiosis 1. So this has to come here. Again, DCE AB. So no answer. All kilada. Can you write, people? DCE AB is a possibility. DCE AB is a possibility. It has to be DCE. Ne? It has to be DCE. Question wrong. Question wrong. Right, question number 14. Question number 14. No further arguments. Ne? Question number 14. Which of the following is correct regarding allosteric regulation of enzymes? Right? Which of the following is correct regarding allosteric regulation of enzymes? Allosteric regulation of enzymes. These are made up of one or more subunits. Which of the following is correct regarding allosteric regulation of enzymes? These are made of one or more subunits. They are asking correct one. These are made up of one or more subunits. Regulatory molecules affect only the shape of an enzyme. People, in allosteric regulation, what is the statement they have given? Allosteric regulation. This is what they have mentioned. Most enzymes regulated by allosterically are made from two or more subunits. Two or more. Our question has one or more. Regulatory molecules affect only the shape of the enzyme. Wrong, the shape and the function. The activity can be activated or inhibited. By binding one substrate molecule to an enzyme. By binding one substrate molecule, the activity can be activated or inhibited by binding one substrate molecule to an enzyme. Is that correct? What it says, people? Activity can be activated or inhibited by binding one substrate molecule to an enzyme. Cooperativity, we can see that. Substrate go and bind and then activate the rest of the active sites. Substrate go and bind. So I feel third one is okay. Regulatory molecule bind to specific regulatory site of an enzyme irreversibly. Regulatory molecules are reversible. So irreversibly is a wrong statement. Intermediate products of metabolism involves in regulation of the products of more end products than the required. Intermediate products of metabolism involves regulation of the products. Intermediate the end products. The end product. Third one is the answer. Third is the answer for question number 14. Question number 15. The effect of certain factor on the rate of the enzyme reaction is shown in the following graph. Which of the following factors could be denoted by x-axis? X-axis. This is x-axis, this is y-axis. pH. pH is correct. Substrate concentration. Enzyme concentration. So pH is good. 15th one, people, first one is the answer. Question number 16. Photorespiration is a process of consuming carbon dioxide and releasing oxygen. Photorespiration is a process of consuming carbon dioxide and releasing oxygen. Consuming oxygen and releasing carbon dioxide. Wrong statement. Take place only in C3 plants. Photorespiration happen in C4 plants, but repress neither. Repress take place only is wrong. Happen in C4 plants also, but greatly repressed. That is what the resource book says. Right? 16th question. Right? And then people is useful process to the plant. No, neither wrong. An energy generating process, wrong, neither. Energy consuming process occurs only in mitochondria and peroxisome, wrong, neither. Chloroplast also. Now we have a problem. Now we have a problem. What is the answer? Now all statements are having certain errors. Right? Now you have to find people the best answer. What is the best answer? Can you suggest me the best answer? You say fourth one is the best answer. Fourth is the best in a generating process. I think second is the best answer. Second is best. Why? Because take place only in C3 plants. We know that it's greatly repressed, greatly repressed, right? So greatly repressed, great, not repressed also, greatly repressed. Even though it produces 3-PGA. What that means, right? So therefore, I think people, second is the best answer here. Right? No good answer, but the best answer you can end up with the second one. Any arguments on that, people? Any arguments, my dear students? Namidu says, even though it produce 3-PGA, even though it produce 3-PGA, even though it produce 3-PGA. What is the point then? What's the point? What is your argument? I cannot understand that. Question number 17. Which of the following statement is correct regarding C4 photosynthesis? C4 photosynthesis, people, correct statement about it. First stable product is 4-carbon compound. First stable product is 4-carbon carbohydrate. Carbohydrate is wrong, neither. Oxaloacetic acid is not a carbohydrate. Then the second carbon fixation is catalyzed by PEP carboxylase in bundle sheet cells. PEP carboxylase is doing it for the first time, and it's present in the mesophyll cell, not in the bundle sheet cells. Wrong. That's wrong. Mesophyll cells abundantly having peroxisomes for photorespiration. 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 Peroxisomes, no, neither. Mesophyll cells abundantly having peroxisomes for photorespiration. Mesophyll cells abundantly having peroxisomes. What we said, people? What we said? Photorespiration is present in C3 plants, not in C4. I mean, up to a certain level, it can happen. Mesophyll cells abundantly having peroxisomes for photorespiration. C4 plants, wrong, C4 plants, not correct. There are many adaptations found in efficient carbon fixation at high concentration of carbon dioxide. There are many adaptations found for efficient carbon fixation at high concentration, low concentration of carbon. Wrong once again. It was a light dependent reaction take place in the mesophyll cells. Light dependent reaction take place in the mesophyll cell. That is correct. In C4 plants, light dependent reaction happen in mesophyll cells. Answer is the fifth one. Fifth one is the answer. Fifth one is the answer. Right, question number 18. Question number 18. Question number 18. Question number 18. Which of the following statement is false? Which of the following statement is false? Right? Which of the following statement is false? chlorophyll molecules, other organic molecules, and proteins are organized into complexes called photosystems in the thylakoid membrane. I'll take it as correct. Correct. In cyclic electron transport, ATP is produced but not oxygen. Correct. O2, O2. Cyclic electron flow occurs only in photosystem 1. Cyclic electron flow occurs only in photosystem 1. Cyclic electron flow occurs only in photosystem 1. That is also correct. P700 chlorophyll, a molecule of photosystem to absorb light at 700 nanometer wavelength effectively. P700 chlorophyll, a molecule of photosystem to absorb light at 700 nanometer wavelength effectively. P700 chlorophyll, a molecule of photosystem 2 is wrong, neither. That is, photosystem 2 has P680. NADP plus is reduced into NADPH at photosystem 1. Not wrong, correct, neither. So we'll take people fourth one as the answer. For question number 19. Question number 19. The above graph, we can't see the graph. So shall we make it a little smaller, neither? The above graph, even if you say above graph, the graph below, the below graph. The above graph shows the rate of reaction versus substrate concentration. Select the limiting factor for the alphabet in the graph portion of the graph. Right. What is this graph, people? You have to identify the graph. The above graph shows the rate of reaction versus substrate concentration. This side is substrate concentration, right? This is substrate concentration. This side is rate. Rate. Select the limiting factor for the alphabet in the graph A, B, C, D. A, definitely A, enzyme concentration. Substrate. Oh. So this part is limiting. Something is limiting here. Remember, something limits. Then after this point, what is actually limiting here? Substrate is limiting. Beyond this point, the point that I circle now, beyond that point, substrate is no more a limiting factor. So substrate concentration is a limiting factor at A. So enzyme concentration, wrong. Enzyme concentration, wrong. So 19th question. First two we can reject now. B. What is the limiting enzyme concentration can be limited after B? Beyond B, people. Beyond B. So beyond this point. Right? Beyond that point. Substrate is no more limited. So probably enzyme can be limited. Enzyme can be limited here. So enzyme, substrate, enzyme. C is, what is C, people? C. C is when the substrate concentration increases, inhibition disappears. High substrate concentration, no inhibition. Can you understand that? High substrate concentration, no inhibition. Inhibition disappears. Now let's take this. If inhibition disappears in high substrate concentration, look at this, people. In the graph, this is substrate concentration this side, right? This is the rate. In one kind of enzyme regulation, when the substrate level is higher, when the substrate level is higher, the inhibition disappear. What is that inhibitor? Competitive or non-competitive? Very high substrate level, inhibition disappears. What is that? Competitive or non-competitive? What do you say about that, people? Competitive or non-competitive? Come on. Competitive. How is that? Higher the substrate concentration. Now we learned that are musical chairs, right? The boys and the girls team. Lot boys are the substrate. Boys are the substrate. Girls are the inhibitors. Girls are inhibitors. Girls are inhibitors. Now, more boys present in this, the chance of boy to win this contest is higher. More boys are there, the chance for the boys to win this contest is higher. So when you increase the substrate level, inhibition decreases. Right? What do you call that? Competitive. So look at this, people. You increase substrate like this. This is the normal graph. This substrate content is sufficient for the maximum reaction. Let's take 10 substrate is enough. When there is a competitor present, right? When there is a competitor present, right? You need more substrate. You need to add more substrate to overcome the inhibition. So more substrate. So very high substrate level, the normal rate can be obtained. What is the C graph, people? What is shown by C graph? Competitive or non-competitive inhibition? C graph shows you the competitive inhibition. So competitive inhibition, this one. D has to be non-competitive. D has to be non-competitive. So the answer is going to be third. This is a little tough question. Tough question. Right? In non-competitive inhibition, the inhibitor damage the substrate. This girl go and break the chair. Right? She slice it. Break it into pieces and then use them as firewood. The inhibitor. So is there any musical chairs now? No chair for the competition. No chair for the competition. So that can happen. That can happen. All right. So what happens there, people? What happens there? The non-competitive. Non-competitive does not mean, people, it doesn't matter whatever the substrate available. If this one lady is there, she destroys the whole event. Right? She destroys the entire event. So no competition. Right? So that sort of an inhibition, when you increase the substrate, it doesn't change it. Right? Reaction rate drop down, people, it doesn't change. It doesn't change. It doesn't change. You can't alter it. So this is a little out of our discussion limit. But anyway, people, we can take third one as the answer. Are you happy with the answer, people? Are you happy with the answer? Right? Moving to next question, question number 20. Question number 20. Question number 20. Which one of the following statement is correct regarding anaerobic respiration? Following is correct regarding anaerobic respiration. Check that. In ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. In ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. Now, neither. What is the hydrogen acceptor there, people? What is the hydrogen acceptor in ethyl alcohol fermentation? Come on, tell me. Ethyl alcohol fermentation, acetaldehyde. Acetaldehyde. Remember that, neither. Pyruvate converts into acetaldehyde. Acetaldehyde becomes this one you remember, This one you remember, right, people? The ethyl alcohol fermentation. and lactic acid fermentation, we learn difference. Glucose becomes pyruvate. Glucose become pyruvate. Pyruvate become lactate or lactic acid. Pyruvate become lactate, ne? Or it become acetaldehyde. Acetaldehyde, and acetaldehyde becomes ethanol, ne? Acetaldehyde become ethanol, right? Acetaldehyde become ethanol. So here the NAD plus becomes NADH, right? So that the hydrogen accepted is acetaldehyde. Lactic acid fermentation, this hydrogen is accepted by pyruvate. That hydrogen is accepted by pyruvate. So if you take that, which of the following ethyl alcohol fermentation primary hydrogen accepted is pyruvate? Wrong. End products of lactic acid fermentation are lactic acid, ATP and NADH. NADH NAD plus. NAD plus lamai. Can't be NADH ne. End products of alcohol fermentation is carbon dioxide, ATP, NADH. Wrong. ATP is synthesized when pyruvate is converted to lactic acid. ATP is synthesized when pyruvate is converted to lactic acid. Wrong. Lactic acid. Wrong. Then hydrogen released from one molecule of glucose reduce 2 NAD plus into 2 NADH. Hydrogen released from one glucose molecule reduce 2 NAD plus into NADH is correct. Fifth one is the answer. Right. Question number 21. Question number 21. You have to now select the answer based on this key. Select the correct statement regarding observations given below. Select the correct statement regarding the observations given below. Color of iodine is converted to blue-black color by starch. Reducing sugar gives brick red precipitate in Benedict test. Protein turn into purple color when heating up with biurate reagent. When fat is mixed with water, the system shaken well, colorless solution was obtained. Amylase add the starch solution after some time will answer to iodine test. Check that, people. Check that. Color of iodine is converted to blue, black by starch. What do you say about that, people? Correct, neither. Ooh. That is correct. Right? That is correct. People, my key is wrong. This is ABD, first one. ACD, second. There is a small error here. ABD, I think it's wrong in your one. ABD, first one. Combination. Color of iodine is converted to blue-black color by starch. Hurry. Reducing sugar gives brick red precipitated with Benedict's. Ekat hurry. Protein is turned to purple color when heating up, boiling with biurate reagent. Biurate reagent. Purple color when heating up. Protein is turned to purple color when heating up to boiling. Heating up to boiling with biurate reagent. Boiling, not boiling kilagone. Wrong kilagone. Boiling not necessary kilay. When fat is mixed with water, the system is shaken well, a colorless solution was obtained. We didn't discuss anything like that. Amylase is added starch solution after some time will answer iodine. Amylase added to starch solution after some time will answer to iodine test. Does not answer neither. Amylase is added starch breakdown. So iodine, no starch available to answer for the iodine test. We discuss a question like that. Right? With the porcelain tile, the white tile using that we transfer drop cut every 2 minutes in that experiment. So people, AB must be the answer. AB is the answer. Question number 2. Question number 2. Which chemical reaction take place in bundle sheath cells of C4 plants? Which chemical reaction take place in the bundle sheets of the C4 plants? Bundle sheet of the C4 plants. 3PGA to G3P. Bundle sheet කියන්නේ ඉන නේද? Bundle sheet 3PGA to G3P. 3PGA to G3P, will take it as correct. 3PGA become 1,3-bisphosphoglycerate and then become G3P, correct. REBP, carbon dioxide, become 3PGA, happen in bundle sheet, correct. And PEP plus carbon dioxide oxaloacetate does not happen in the bundle sheet, that happens in the mesophyll cell. Malate convert to pyruvate, that happen in the bundle sheet. Oxaloacetate converts to malate. Oxaloacetate convert to malate. That happen in the where people? That happen in the mesophyll cell. That happens in mesophyll cells. So ABD combination can first answer. ABD combination first answer. Is that okay? Right, moving people to the last set. Moving to the last set of questions here. 9 o'clock. Select the molecules produced during the photosynthetic pathways. Select the molecules formed during the photosynthetic pathways. Right, acetyl coenzyme A. During photosynthetic pathways. Acetyl coenzyme A. Select the molecules produced during photosynthetic pathway. Acetyl coenzyme A, wrong. Neither. Where do you get acetyl coenzyme A? Cellular respiration. Acetaldehyde, cellular respiration. Oxaloacetate, correct. Pyruvate, correct. NAD plus, wrong. Enang C and D must be the answer. C and D must be the answer. Question number 24. Question number 24. Which of the following are true regarding interface of eukaryotic cell? Following are true regarding interface of eukaryotic cell. The cell growth occurs in G1 and G2 phase. Cell growth occurs in G1 and G2 phase, correct. 24, first statement is okay, ne? It was a histone protein are synthesized in the S phase එකත් හරි නේද? Number of chromosomes in the cell become double at the end of the interface. Double at the end of the interface. Number of chromosomes. The DNA double in the end of the interface. Wrong statement. DNA content of the cell increases only in the S phase. DNA content of the cell increases only in the S phase, correct. Right, and increasing of the number of the organelles happen only in the G1 phase. Increasing of number of organelles happen only in the G1 phase. Increasing of number of organelles happen only in the G1 phase. Wrong, G2 phase also. So we can take ABD answer once again. Question number 25. Last question. Question number 25, last question. Now, people, you answer the question and then you upload the paper, right? You answer the questions and upload the paper. Our personal folders will upload. So we'll correct them and upload your answer sheets to the personal folder, right? So isn't that the false that the 3PJ convert to 1,3-bisphosphoglycerate, not to G3P? No, that's correct, but it's like a summary. Now there are so many other intermediates also, right? 3PGA converts into G3P finally, right? Because now, right? Because you also 1,3-bisphosphoglycerate also a summary, there can be some more intermediates, right? So we don't talk about that, right? People, question number 25 in feedback inhibition. In feedback inhibition, the feedback inhibitor. In feedback inhibition, the metabolic pathway is stopped by the inhibitory binding of its end product to the enzyme. In feedback inhibition, metabolic pathway is stopped by inhibitory binding of its end product to the enzyme. I'll take it as correct. To the enzyme, one of the enzyme in the pathway. This limit the production of more end products than required. Correct, When activator bind with regulatory site, stabilizes the shape with functional active sites. When the activator bind with regulatory site, stabilizes the shape with functional active sites. Correct. Correct. Feedback inhibition is an essential process regulate end products produced in metabolism. Feedback inhibition is an essential process. Regulate the end products produced in metabolism. Feedback inhibition is an essential process, regulates the end products produced in metabolism. Feedback inhibition is an essential process, regulates end products produced in metabolism. Feedback inhibition is an essential process, regulates end products produced in metabolism. In the case of ADP exceed limit, catabolism slows down as ADP molecules function as allosteric inhibitor. In the case of ADP exceed the limit, in the case ADP exceed limit, catabolism slows down as the ADP molecule function as allosteric inhibitor. ADP is not an allosteric inhibitor neither. It is an allosteric activator. So I'm taking A, B, C, D as the answer. A, B, C, D as the answer. A, B, C, D as answers. Right, people? Some more are there to discuss. Right? So, the structured questions. Now the time is over, people. Structured will take into another class discussion. I'll give you the marking scheme. You upload the paper. And then we'll discuss people the marking with you. Hari. You upload the paper. Hari. It was say we'll discuss it. Otherwise, it's going to take. I'll give you the marking scheme anyway on Saturday's class. I'll give you the marking scheme, right? So then we'll stop the discussion from here, right? We'll stop the discussion from here. I'm giving you people the next paper. You can paper number paper 5 ne. Paper number 5 I'll be giving you people on coming Saturday with paper number 4 marking scheme. And you upload your paper now. Tonight you have to upload your paper. So we'll mark it and then we'll upload the answers there. So you can check against the marking scheme, right? So that's all for the day, people, because this is the allocated time. So we'll try to finish it in the allocated time period. So thank you very much for your participation. Hari, thank you very much for your participation, and I'll see you all people soon in the physical class. I'll meet you in the physical class, and so have a nice time. Right? Good night, and I'll see you soon.